Welcome to Easy Microbiology.
Good morning. Today I will be talking about Acinetobacter hemolyticus, an opportunistic pathogen that can be found in various clinical settings. I will explain the potential implications of this organism in clinical practice and discuss the best possible strategies for prevention and control.
Acinetobacter hemolyticus is a species of gram-negative bacteria. This means that the bacterial cell wall is composed of a peptidoglycan layer and an outer membrane. It is an opportunistic pathogen, meaning it typically infects individuals with a weakened immune system. Although research on this particular bacterium has not been as extensive as Acinetobacter species, it is still being identified as a potential pathogen. Acinetobacter hemolyticus is a microorganism commonly found in soil, water, and hospital settings. It has been identified from clinical samples and has the potential to cause infections in healthcare settings, making it an opportunistic pathogen. Acinetobacter hemolyticus is a non-motile, rod-shaped bacterium, typically found in single cells or pairs. Its cell wall contains lipopolysaccharides, making it a pathogenic organism. Laboratory testing through media such as blood agar or McConkey agar can be used to culture and study this bacterium further. Acinetobacter hemolyticus is a pathogen of the Acinetobacter genus, which is an opportunistic pathogen. Culturing this pathogen on common laboratory media such as blood agar or McConkey agar will result in smooth, round, opaque colonies with a creamy or pale color, distinguishing it from other similar species. Acinetobacter hemolyticus, an opportunistic pathogen belonging to the Acinetobacter genus, is oxidase negative, non-lactose fermenting, and does not produce gas from glucose fermentation. Additionally, it is catalase positive, meaning it produces the enzyme catalase which breaks down hydrogen peroxide. Acinetobacter hemolyticus is an opportunistic pathogen that primarily affects humans, particularly those with pre-existing medical issues or residing in medical settings. It has been detected in clinical specimens from blood, wound swabs, and respiratory samples. It is noteworthy for its broad host range, capable of infecting humans, other mammals, fish, plants, and some invertebrates. Though it is not a significant human pathogen, Acinetobacter hemolyticus can still be responsible for dangerous and occasionally fatal illnesses when it gains access to human bodies.
Acinetobacter hemolyticus has not been extensively studied in terms of its virulence factors compared to other Acinetobacter species. Nonetheless, it has been discovered that the bacterium has outer membrane proteins and lipopolysaccharides that could play a role in causing disease.
The bacterium Acinetobacter hemolyticus has the ability to develop antibiotic resistance, which may lead to treatment failure. This resistance is achieved via mechanisms such as the production of beta-lactamases, efflux pumps, and alterations in membrane permeability. Of particular concern is the ability of this bacterium to acquire resistance genes through horizontal gene transfer, making it resistant to multiple antibiotics. Acinetobacter hemolyticus is an opportunistic pathogen that is commonly found in hospital settings and other healthcare facilities. It has been isolated from clinical samples on numerous occasions, suggesting it has potential as a nosocomial pathogen. Studies into the epidemiology of Acinetobacter hemolyticus have been limited, compared to other species of the Acinetobacter genus. Acinetobacter hemolyticus is associated with numerous infections, ranging from bloodstream infections, respiratory tract infections, to wound infections. It is notoriously hard to eliminate due to its strong capacity to invade host tissues and create biofilms, thereby allowing it to cause persistent, chronic infections. Immune responses are essential for treating Acinetobacter hemolyticus infections. Neutrophils, macrophages, and cytokines have a role in eradicating the infection. Nevertheless, the precise immunological reaction to this bacterium is still yet to be fully understood. Researchers are continuously investigating this issue in an attempt to develop enhanced treatment approaches. Diagnosis of Acinetobacter hemolyticus infection can be made by isolating and identifying the bacteria from clinical samples. Culturing techniques on appropriate media and biochemical tests can be utilized to do this, or molecular techniques, like PCR, can be employed for more rapid identification of the pathogen. Proper prevention strategies are essential for ensuring patient safety from Acinetobacter hemolyticus infections in healthcare settings. Hand hygiene, disinfecting of surfaces, and implementing isolation protocols are the primary methods of prevention. Adhering to these practices can greatly reduce the chances of this pathogen's transmission. Patient education on the prevention of infections should also be included in the strategies. Antibiotic resistance is a major concern when it comes to Acinetobacter hemolyticus infections, posing a challenge for successful treatment. In severe or resistant cases, a combination therapy or alternative treatment options may be necessary. The selection of antibiotics must be determined by susceptibility testing in order to generate the most optimal outcome. It is evident that Acinetobacter hemolyticus is a pathogen of particular concern in healthcare systems. Its virulence factors, mechanisms of antibiotic resistance, and immunological responses are all topics that require further research. I greatly appreciate your time and attention in listening to this presentation.